Okay, so I will kick off. So good morning, I'm Salome, and I'm one of the Vax-Hub researchers, and it is my pleasure to welcome all of you to the live MiniTEM webinar in collaboration with Vironova. Before we start the session, I'd like to remind you that it's been recorded. So for those of you who have problems with that, please let me know so that when make it public, you will be kept anonymous. Also, during the session, I would like to ask you if you can keep your microphone and webcam off to avoid any connectivity issues. Oh. So this session will cover how the MiniTEM can effectively be used for sample characterization, and this will be shown by a live demo performed by our host, Dr. Saba, who is an application manager and senior scientist at Vironova. And Saba will be using Adeno-Associated Virus as a case study to demonstrate this. Afterwards, there will be a Q&A, and for this part of the session, I would like you to ask either to raise your hand or type your question on the chat of the webinar. Because we have time constraints, we might not be able to answer all your questions, so please either leave your questions on the chat or send us an email and we will answer them as soon as possible. Quality assessment is of paramount importance in our area of research and the only guarantee to meet the regulatory requirements and reach the market. For that, we need robust analytics to be able to evaluate properly our samples, and an example is the biophysical characterization, which we'll be covering during this session by using the MiniTEM system. This system, in comparison with the standard TEM, is more user-friendly, contributing in a certain way to a faster way of assessing the quality of the analyzed samples. The VaxHub has been collaborating with Vironova for a couple of months, and recently an interactive voucher was awarded to the project entitled VLP and Viral Vector Quality Assessment. The researchers leading this project are Steve Morris and myself. So for those of you that wanted to know more details about the project or that are interested in using the MiniTEM system, please drop us an email. Also, in the following days, you will receive an email with a feedback form that we highly appreciate if you could answer and return it to us. The purpose of this form is not only to know how and where we can improve our webinars, but also to know who among all of you would be interested in participating on part 2 of this webinar. We aim that part 2 will be completely practical and it will be tailored to your image analysis requests. You don't need fresh samples to participate. An alternative to that would be EM images that you might want to analyze again. Now, without further ado, I will hand it over to Saba. Saba, the webinar is yours.
Thank you. Thank you so much. I'm really excited about this webinar. I think this is going to be great fun. And I hope you guys can bear with us if we have some technical issues, but hopefully we'll all run smoothly, but let us know if there's anything that is sort of acting up. As I said, as Salome mentioned, my name is Saba Hussein-Gore. My background is in virology. I've also worked quite a lot on membrane proteins, so I've done a lot of purification, expression and purification using a whole range of different cell lines, and I've also got experience in microscopy. And using the MiniTEM, and for this purpose, those three, that background has really come in handy. And I want to start off with just a really quick presentation, just a couple of minutes, Can someone see my presentation just so that I get the...
Yes.
There, actually, I can see that I can see my presentation. Okay, so I'm just going to spend just a few minutes giving you a quick overview, just preparing us for the imaging session that we will be doing. So first off, I'm just going to tell you just a little bit about Vironova, who we are and what we do. I'm going to then give you a very quick introduction to TEM and its applications. I will just introduce you to the MiniTEM system, both the hardware and the software. I will give you an example of one of our software features, which is an AI feature called CNN. That will then lead me on to our COVID-19 response, and then we'll start the imaging. So who are we here at Vironova? So Vironova is a Swedish biotechnology company. We've been around for about 15 years, and we are providing comprehensive hardware, software, and services for the analysis of nanoparticles. And we do this really by opening up access to transmission electron microscopy and transmission electron microscopy based image analysis with the biopharmaceutical development in mind. The application areas, our main application areas, are in the field of gene therapy, vaccines, and drug delivery particles. And the kind of analysis that we can provide for these application areas are looking at, for example, a particle size distribution in your sample. You can look at the purity of your sample, how clean it is, and compare it from different stages of purification. You can look at the integrity, so how intact is your, let's say, your viral vector. We can also look at some structural details and look at some morphological features on slightly larger viruses, like let's say lentiviral vectors. We can also do full empty analysis using the cryo-EM method. And then we can do thin sections, so we can look for retroviruses in production cell lines. So this analysis that we do, it comes from images that we acquire through transmission electron microscopy, TEM for short. And I think maybe a lot of you are very familiar and users of TEM, but for those who aren't, I've just got this one slide and I just want to talk to you about TEM just very quickly and just describe to you what it is. And I think the easiest way to explain that is to compare it with a light microscope. So in the light microscope, you typically have a light source and it's applied to your sample. And you have different objective lenses that allows you to visualize your image, often through an eyepiece. With TEM, the difference is that we are doing this with, so instead of a light source, we are doing this with an electron gun. And rather than having our lenses are actually electromagnetic lenses. And that allows us to visualize our image, and we look at it through a CCD camera. Our sample is on a grid, and I will show you a grid later. And it's been stained with heavy metal salt just to increase the contrast. So what happens is that when your electrons go through, they pass through areas that are less dense, and the areas that are dense are absorbed. So that's how you can generate your image. And the MiniTEM, it does the same thing, but just in a smaller format. It's more compact, and it's also a low voltage electron microscope. So normally these electron microscopes are about 100 kV plus, and the MiniTEM is about 25 kV. And the images that we get, so why do we think that they're so valuable? I think the value really comes from that it's a direct analytical method. So you can get some really good insight into your sample. If you want to know how big your virus particle are, you simply measure it. I'm going to use my mouse. I don't know. Oh, no, I'm not. Like this. Okay, fine. I'm pointing. There are some images, and I'm referring to them. This is just an example of what we're comparing two different methods. So the MiniTEM, which is our direct analytical method, or the TEM imaging, and we're comparing this with dynamic light scatter, which is sort of an indirect method, looking at the movement of particles in a solution, and taking out the sort of size information from that. The sample that we are looking at here is an AAV sample. So what you see with yellow circles around, those are actually AAV particles. And The smaller circles, I'll get to them in a second. But if you look at your graph to the left, you can see that with the DLS, the size distribution appeared as a pretty broad peak, ranging from between 20 to say 60 nanometers in size. But when we did the analysis with the MiniTEM, we could see that we had two different types of particles in our sample. And so we could sort of dissect this broad peak and see that it was actually composed of two different types of particles, our AAVs, which I mentioned, but it also had a host cell contaminant. In this case, it was proteosomes, which are about 14 nanometers. So just an example to show you what we can do. And these two images, one of them is a sort of more magnified image of the one next to it. Another example is that you can start comparing your purification techniques. And with most analysis methods, of course, they do well when you just use them to measure one thing, but they do really, really well when you want to compare two different things or three different things, when you want to know which direction to go in your downstream processing or in your purification technique, or which type of column you want to use. In this instance, here is a viral vector that was expressed using baculoviruses and insect cells. And the first step of the purification, there were two different options to take. One of them was to use affinity chromatography, and the other one was to use sucrose gradient. And we can see from our TEM images that in our affinity chromatography, I want to point with a pointer, but I can't because but anyway, the picture to the left shows that you can actually still see your baculovirus in your sample that wasn't purified out, whereas in your sucrose gradient, we couldn't detect any the presence of baculovirus. So it helped to take the decision that the sucrose gradient would be a better option for this purification step. And we can, of course, look at a whole range of particles. I just wanted to show you a quick example of some of the things that we have imaged and that we do image on a regular basis. So we can look at non-enveloped viruses here. To your top left, you can see like adenoviruses. We can look at envelope viruses, so pleomorphic virus particles. This is influenza in this image, and you can perhaps, if you look closely, you can see that there are some spikes that you can detect. We can look at smaller things like larger recombinant proteins. And of course, we can't get the same kind of detailed information from them, but we can look at their particle size distribution, and if we're detecting any aggregation or something in the sample, we can look at things like lipid vesicles. We can look at drug delivery nanoparticles. We can look at tissue sections, so thin sections, and look for retroviruses. And here in the bottom, this is sort of one of our smaller particles. We can look at gold nanoparticles as well, and vaccine adjuvants. And then when I did this presentation, I saw that actually I didn't have bacteriophages in there, so I put that in. So we can also look at bacteriophages, which I really like looking at. I think they look really nice and cool, and we can do some very nice measurements on bacteriophages. So a whole range of samples can be imaged with TEM. And this imaging, the MiniTEM system, just really quickly. Thank you. It's a low voltage TEM, as I said. The optical particle size that we like to look at is between 10 nanometers and 15,000 nanometers. It's got a resolution of 1 nanometer, and it doesn't really require an expert user to use it, so it should be easy to use, and you don't need any specific facility, so you can just plug it in a single wall socket and run it. The software that is part of the MiniTEM system is called VIAS, which stands for Vironova Imaging and Analysis Software, and it controls the microscope, and it also provides us with this semi-automatic image acquisition as well as analysis and data presentation in this. And you will be more familiarized with this as we do our webinar and our live imaging. Just a really, really quick note. One aspect of our VIAS software is that we have got CNN model in there. So CNN stands for convolutional neural networks, and it's a form of artificial intelligence. It's kind of inspired by biological processes in the animal visual cortex. For me, what I sort of understood from it is that basically you give it a set of data that will work as your trainers, and you tell it to, you do an annotation on it. So you don't need to explain the characteristics, you just tell it what it is. And you do that with a couple of images, and then once it's been trained, you can then train it on a much smaller subset of images, and it can do the classification. So we have done the base training on thousands of expertly annotated TEM images, and it means now that we only need 10, 15 images to do new annotation that we manually annotate, and then we just let it run, and then we can run a nice particle characterization. And I'll show you how the CNN runs in our live imaging session. But we think it's a very powerful tool, and it's really powerful as well that we can train it so quickly. So this kind of leads me on to the COVID-19 episode. When the pandemic started, I think we all sort of thought, okay, well, what can we do? What can we provide? And we instantly knew that making VS accessible and making CNN more accessible and training our CNN could be useful. So this is how we support the fight against COVID-19. All of these options are free. We don't want to charge, we just want to help out. And our options are to make our system available to you guys. So there are just 3 quick options. One of them is if you have grids already, you can send it to us and we can image it for you. The second option is if you don't have the capabilities of preparing a grid, you can send us your sample and we can prepare those samples for you and image them here in Stockholm. And finally, which is probably the easiest one, is if you already have taken images with an electron microscope, you can send us these TIFF files and we can run them on VS for you. And this is not just something that we're just doing because we want to sort of jump on the bandwagon. We really are passionate about this. We care about this. I just want to show some pictures that my son and the kids in his preschool prepared after I went and spoke to them about how important it is to develop a vaccine, how important it is to wash your hands. I mean, this is something that we really want to help out with in any means possible. So please, if you feel like you have something that you might want to analyze, do contact us or Salome, and let's see how we can help you. So on that note, I am finished with my presentation, and next up we will do the live imaging session. From the very start to the finish of the session, or the entire process, should I say, from when you have a sample in a tube until you have your statistics and your data, should take 75 minutes. But I have done the sample preparation beforehand so that we can get started straight away. We have sent out a link where there are videos that shows how the sample preparation is done. So please have a look at them. If you haven't looked at them now, you can have a look at them later. They are still on there. I just want to show you very quickly what a grid looks like. So when we prepare our samples, we prepare it on a grid. I can just show you really quickly what it does. So we have a grid. Can you guys see me in the video as well? Yeah?
Yeah, right at the bottom.
Okay. Right. I can't see myself, but I hope, can you see this grid?
Put it a little bit further to the left because you have some white space.
Like this?
Just a little
You're
bit. Yeah, a little bit to the opposite direction.
Oh, good. Okay. Oh, this is gonna. Okay. Okay. Hang on. Like, oh. All right. Oh, this is really hard. This is a grid. I guess. Okay. Look, I'll do this. I'll put it next to my face so you can see. It's really small. It's very small. It's very thin. These are the grids. This is a 400 mesh grid. And we prepare it really by placing 3 microliters of our sample on there. We wait for a minute, we blot it off with some filter paper, then we do 2 washes with 3 microliters of water, blot it off with a filter paper as well, and then in the end we put our stain solution on. We leave that for about 10 seconds, and then we blot it again with our filter paper. We leave it for a little bit to dry, and then once that's done, we put it back into our grid box, and it can be left at room temperature and it can be stored for, we say a year because the grid itself can sort of start break a bit, or the carbon support, but you can have it for longer than that. So you can image something, you can re-image it again. But that's it. So that is what we did. And now we're going to do the imaging. We'll do the image analysis, and then we will do the interpretation of the results. So I will now stop this presentation, and let me see now. I'm going to stop presenting this, right? And I will now go to share here, right? Can everyone see the screen that I am sharing?
Yes. Yes.
Good. Okay. Right. Was there any questions during the presentation? Thank you.
Is it possible to make this screen larger?
This screen?
Yeah, is it possible? If not, no problem. But if it is, it would make it clearer for me because I can't see very well. But that's because of the way I'm running the actual Teams.
Yeah. Okay, so I think this text, this is probably going to be the smallest text you can see. I will bear that in mind. I will make it. I will try to zoom in. You don't need to read any of this text here, but thank you for pointing that out, and I'll make sure that I zoom in some of the areas. If you think that it looks too small, give us a little heads up. This is unfortunately as big as I can make it, as it's a full screen on mine, but I'll bear that in mind. Thank you. So if there weren't any questions, I will get started with the imaging session. So I want to just really quickly draw your attention. So when we do an imaging session on the MiniTEM, we use something called our imaging session instructions. It's a single sheet. It's normally laminated, and it gives you all of the steps right from the start to finish. So we will be using this as our aid to do our imaging today, but I'd like you to see. So step one basically is start the microscope with pressing the mic button, and the final step here is finally switch off the mic button. I keep looking at you when you're there. So I have opened the VS software, and this is what it looks like. So we have 4 panels. They're called Start, which is where we are now. We have imaging, analysis, and workflows. The Start panel, so the VS software is actually run on a global database, which means that you have some resources that you can share as long as you give access to this database for the users. They can access those, for example, CNN models if you want to share them, and your classification classes. When you open it up, you have a list of your projects here, and you can just click Start, Create New Project. I've already created a project because I wanted to get that sort of ready, but you would just click Create Project and start it. I've got one here. It's called VaxHub, and I will double click on it to get started on the session. So now that I've started my project, I'm going to go into my imaging panel, or my imaging tab. In my imaging tab, I've got something here called Imaging Control, and this is where I can turn on my beam. So we will start by turning on our beam. But actually, before I'm going to do that, I was actually going to show you what the MiniTEM looks like. So I will click beam on, and while I'm turning the beam on, if you can look at the small screen, I just want to show you what the MiniTEM looks like. Thank you. Just so you get a sense of it, right? Can you? I hope you guys can see this. So this is another MiniTEM here, but so this is the MiniTEM. Och Mats, om du bara kommer hit. If I open this up, this is the bit that we can kind of move and control. So we have two knobs here. This is for adjusting the apertures, and we have a little screw up here. Yeah, that's perfect. To control our YAG screen, which I'll get to in a bit. So those are the only things that you can kind of move and change. So not very much really. And the sample insertion that was also sent to you in the video is here. So this is my grid holder, and I insert my sample through here by opening and closing the airlock and following the instructions on a GUI that tells you step by step how to do it. And during this sample exchange, you have to wait for the vacuum to be created in your what are the column like and that takes a couple of minutes, and you have to do that for each sample exchange. So you need to make sure that you don't lose the vacuum in the column, and that's why you need to do this in a sort of careful step-by-step manner. So if you can see the screen here, we are starting the beam. The beam is, you can sort of track the progress of it. I can show you just a little bit of what this imaging control looks like. So here we can control the magnification of our image. Oh, look, now it's turned on. We can also control things like illumination and auto exposure. Here is where we will acquire images during the session. And to your right, we've got different panels. So right now I've got the histogram up. You can also look at the DFT. And you also have all of the microscope controls here. I don't know where I should put this back. Right here. Right here, we have something called stage control. This is effectively our grid, and we are here in the middle. When I scroll with the mouse, I can zoom in. Is this difficult to see, guys? Maybe this is too small. I'm really sorry if it is. So I am now zooming in, and I am now zooming out. And I can use this to move around on the grid. I can move around on the grid from here if I want to go further distances, or if I want to go to the very edge of the grid, or the middle, or the side. But I can also, in my actual, within the actual image, go to places by clicking. Wherever I click with the mouse, that should go sort of towards the middle of the grid. So this is an AAV grid that we will be looking at. It has been stained with urinal acetate, but there are also other non-radioactive stains that you can use. The urinal acetate is the sort of the most commonly used stain. And you can see I didn't choose like the most beautiful, the very best looking grid in the world. I just put in one of our sort of regular AAV grids, and you can see, I don't know if you can see, but this is what the grid looks like, and you can see that it's ripped at the top. But that's okay, because we have, it's a 400 mesh, so we've got 400 of these grid squares to choose from or to look at. So even if this one is a bit broken, we can always go and look somewhere else. I will start, before I start doing the imaging and start taking images, I will first go to an area where, let's say, where the grid is not broken, and I will start to do some changes to my microscope so that I can get the best images possible. So I'm going to now click on the screen, and when I'm clicking, I'm moving along the grid. This is a grid square, so the dark areas are a grid square. This is looking even less beautiful than the last one, right? I'm going to continue here and see if I can find an intact grid. And sometimes you have this. Sometimes you can find this sort of on the edge more, maybe where you have held it with your tweezers. But it shouldn't sort of make you feel like, oh, this is now kind of impossible. I'm going to go here. Now I'm clicking here, just going to move away. Maybe this bit in the middle is a bit ripped. And see, now you won't feel bad when you have a ripped grid, because you will remember how totally ripped our grid was, which is fine. Okay, let's hope that this one looks good. Okay, this is looking better. This is looking better. Okay, let's use this one. Let's stay here. So at the moment, my magnification here is 23,000 nanometers. And if I click here on the drop down, we can go as zoomed out as 32,000 nanometers. One of these grid squares is, I think, 42,000 nanometers. So when I'm at the most zoomed out, I should be able to see most of my grid. Now I'm at sort of at the edge of my grid here. But I will start with using my instructions, and I will start by going in to check that my beam is centered and round. That's basically what I want. So in order for me to do that, I will find, it says find a visible part of your sample that is not blocked by a copper part of the grid. So I'm going to move a little bit away from the copper grid. Okay, so there. And now I'm going to turn the auto illumination off and the auto beam alignment off. And I will start lowering my illumination value. And as I do this, you're going to start seeing that my beam will start becoming smaller and smaller as I lower the illumination value. And I can then see if it's actually centered or not. When we start doing this imaging, normally we try to spread the beam as much as possible to prevent any damage to the grid. So I'm already seeing that it's not quite in the middle, it's sort of going to the left. So I will use my beam centering buttons here to move it to the middle. Like this. I'm going to make this a little bit smaller, and it doesn't have to be, you know, really precise. You want to have it, I think, like this, and I'm happy with this. This is sort of centered. The other thing that you would want to check is if your beam is nice and round. Right now, our beam is nice and round, and you don't have to change this very much, but it's good to keep an eye on. And I can show you what the beam would look like if it wasn't nice and round by changing my condenser and stigmata values here. So I'm going to show you. So if you start imaging and it looks like this, you would want to make it round so that you don't have problems like astigmatism of the image and so on, and you have a nice and even focus and even image. So I have done this round. Now, how much time do I have left?
It's 11:05.
So how much time do I have? Okay, good. So that's it. So our beam is circular and it's in the middle, and that is what we want to do. So we're happy with that. So then I turn my auto beam alignment on, go down to the middle, and I save these alignment parameters. And it asks me, do you want to save them? And I do. So I say yes. Then I can turn my auto illumination on again. And I'm almost ready to start imaging. There is one more thing that I just want to check very quickly, and that is to see if my apertures, which helps to sort of focus the beam better, that we have these actually physical things, if they are really nice in the middle. And the only way that I can see my aperture is if I zoom out. So I hope you guys can see the edge here. Can you see the movement of my mouse? Yeah, that you can see the edge, and that is nice and centered. So I am happy, and I can now start with my imaging session. So I will click Start Session here, and you can choose what session type you want, whether it's for production or non-production. The only difference is if you click production, you have to enter the information here, which says study, sample, and grid. So let's do that. Let's say this is production. So our study, we can say this is for the VaxHub webinar. And our sample is AAV. And the grid, so you can give it the grid position. So you can use the grid name here. So the grid here was grid A1. And then I click Start Session. So now we're ready to take images. I'm going to start zooming in a bit. Right now, we're sort of in the middle of the grid. I'm pretty happy to start imaging here, so we can do that. But as you saw in the beginning, there was an area where the grid was ripped. you can move away from it. And we also have an educational compendium which shows you examples of what good staining or what a good grid square should look like. And what we are looking for is something that is evenly stained. It's not too dark. It's also not too dry or too bright. It's this sort of nice shade of gray. And we have image examples for you guys to look at to make yourself familiarized with what is a good grid square. A little sort of FYI is that we will have a feature incorporated into our software where we can give it an image of a grid square and we can tell it to run an analysis and then just pick out the grid squares that match the one that we have said for it is like the ones that we want. So then we can do the imaging in the grid squares that we have already predefined. So I'm going to start zooming in and a little more, step by step. You can also go straight down to, you know, a really high magnification. You don't have to do it step by step. I think it's maybe more fun for you guys to start seeing how we're going into the image and what we're starting to see. So I hope that you guys can see that we're starting to see some dots, some bright dots. And these bright dots are actually our AAV particles. Now, you might have seen that something here changed, and what changed is that when we're going, there is a shift in our field of view. As we're going higher into magnification, we need to change the light objective from 4x to 20x, and it just does that automatically. And it sits on a carousel, which is why you see this sort of movement. So I will zoom in a little bit more. I can tell you, we like to look at AAVs at a field of view of 1,500 nanometers. So AAVs are about 28, 29 nanometers, and when we look at them on the MiniTEM, I think their sort of regular size is about 25. So between 25 and 28 is normally what we see. So I'm going to go one more. So this is the kind of magnification that I want. Right now, actually, it's looking like it's quite well in focus, but if it wasn't, so sometimes you need to adjust the focus. So I'm going to just change the Z coordinate. So the same as if you're doing imaging with a light microscope and you're looking at your cells, you need to sort of change the Z to go into focus. So right now, the image, I've taken it out of focus. So this is, so you can end up starting with something that looks like this. And then you might want to get it into focus. You can do that in different ways. You can change the Z value yourself, so you can move in height yourself, but that can sometimes be quite tricky, and you might need to be a bit of a, you know, expert to know sort of where to stop. But another thing that we have is this button up here. It's called autofocus. So when you click on this, we have an algorithm that should hopefully get us in focus. So now it's performing autofocus. So I'm just going to sit and wait and see. Let it do it on its own. So when you're hearing the sound, so what it's doing actually is that it's jumping, it's taking different Z values, and then it's shifting the beam, and it's doing it again. And then where this crossover of the beam happens, that the Z coordinate there, that's what it assumes to be the sort of the focus. So I'm just letting it do it. I'll just wait. How much time? So it's trying out these different sort of values, and sometimes it takes a bit longer, sometimes it doesn't take as long, sometimes it does it, and there has been a few occasions where sometimes it's not quite gotten there, but on average I think it behaves quite well. But I bet now that I'm saying that it's probably not going to do it, but it is a fairly well behaving autofocus. I get that. So while we're waiting for the autofocus, I just want to talk to you quickly about something called waypoints. So the grid is small, but we're looking at really, really small things, and we're moving around in a really small area sometimes. And it can be hard to keep track of where you have been or where good areas are or where not such good areas are. So one of the things that we have is something called waypoints. So if you look here in the bottom right in the stage control, I can put something called the waypoint. So I basically pin the area where I am that I find of interest. So I can do that while now it's done the autofocus, but I can do that just to finish that. So I go here and I click create waypoint. So now it saves this coordinate to me and I can go back and revisit this. I can move around the grid and I can do that. I don't always have to be at that waypoint to, or in that region to put a waypoint. I can just right click and just put new waypoints. So if you want to do a quick analysis and just scan through your grid very quickly, you can put your grid in and then you can just put out a couple of waypoints that are nice and far apart from each other. And then you can run an imaging which images in those waypoints and you can have a good assessment of what the status of your grid is like. It looks like someone's written a question. Is that correct? Or maybe not. So
Yes, it's Steve.
yeah, okay.
Steve, go ahead, please.
No, no question. Keep going. Sorry.
Okay, it's okay. It's nice to hear your voice. Okay. So I look here and actually now I'm focused. I want to tell you guys, for the people that don't really work with TEM, I just want to tell you why this looks a little bit blurry. So I hope they can see this. So in TEM, actually, I'm going to use a different sticky pen. This one. So right now, so this is our grid, and this is our AAV, right? And now it's done the focus, and it's here now. So it says, yeah, I found the focus. This is my focus. But actually, where we want the focus is this area here, right? This is what we really want to focus on, is sort of the edges of the particle. And this value here is referred to as a defocus value. Oh no, I can see my screen here. That's fine. Thank you. And the defocus value can be calculated, and we can actually put this in the MiniTEM. So if we think back at that presentation that I gave, and I showed you that the electron gun is at the top, and then you have the sort of the CCD camera or the detection at the bottom. The MiniTEM is actually upside down. So in our instance, our gun is actually at the bottom and our camera is at the top. This is just to provide more stability to the MiniTEM. But what it means is that our defocus value normally in normal microscopes, you go minus, and in this instance, because we're upside down, we're going to go plus. So that's what I'm going to do. I'm going to use the scroller here, this manual focus MF, and I'm going to click, and that should give me the defocus number here. So I'm going to something like this. Yeah. So now I think you can see that the image is much sharper. I can see that there is a little bit of blurriness here in the top corner, and I'm going to adjust my YAG screen. My YAG screen is basically what converts my electrons into photons before it goes into the camera. And sometimes you just need to adjust that. So I'm just going to turn the screw a little bit, and you can maybe, if you look closely, you can see that there's like a veil of blurriness, and you just kind of have to turn it. Thank you. Yeah. Oh, there's a question. It says, hi, sometime I found that the sample film broke down. So this is a question I'm just reading out. Sometimes I found that the sample film broke down when the exposure was too long to the light and if the sample is too concentrated. Does this happen with the MiniTEM? Thanks. Yeah, so breaking your, so thanks for that question. It's nice to know that like someone is listening, it's great. So if you have, if your beam is really concentrated, you can burn your grid squares, and we have that. It's some stains as well are also a bit more sensitive to beam damage, I guess is what we can call it. And one of the things that we don't recommend, for example, is to use, there are different types of grids that you can buy, and they have different types of slots. So the one that we have is, as I said, it was a 400 mesh. So even if one of them breaks, as I said, you can hopefully always go to another. But sometimes if you have, this is a grid type that is used in thin sections where you actually just have one really big hole. If you damage this here, then you've basically broken everything. And when we start the imaging, we always make sure that automatically our beam is spread as much as possible, specifically to avoid the chances of damaging the grid or the sample in the grid square, so you don't have this happening. I hope that answered your question. So I guess it happens just as much with the MiniTEM as with other ones, but by having the beam spread out more, you should hopefully be able to avoid it. Right now, I'm focusing on this one area, and actually with urinary acetate, it's a stain that can tolerate these things quite well. So yeah, so if that was okay, I will continue. Yeah, okay. So we are now looking at our AAV sample here. I wonder, can any one of you see any particles that look different? Okay, I'll tell you. So these ones look white. So most of them look white. Actually, I can turn this on. This one down here, it looks like it's dark in the middle. So does this one, and this one looks like it's broken. So we use the stain when we are staining our samples. Particles that are broken are not intact. The stain will seep inside, and we will end up seeing something that has a dark center. So we can classify, in this instance, we can classify AAV particles as internally stained. So it's broken and the stain has come in, and internally non-stained. And these are intact. So this sample is pretty good because actually most of our AAVs are intact, and we only have a couple of AAVs that are non-intact, that are broken. I'm just going to adjust this thing here. Just like that. And we can take an image of this, and we can do some manual annotation of this if we want. So I clicked on this capture, and I can look at my image. I can zoom in so we can start, so we can have a look here. We can measure our particles. Yeah, 24 nanometers. It's the broken one as well. 23 nanometers. So that's the correct size. We can look and see if we have any. Here is some sort of aggregation here in the bottom, if you guys can see that. And I can start doing some manual annotation. So I'm going to transfer this, click transfer to analysis, and I'm going to take this to my analysis, and I'm going to have a look at it here. So this is my analysis tab, and this is where the images get transferred, and this is where we can do annotations. We can also look at the properties of the image. So I'm just going to make this a little bit like this. Oh. Right. This one is a bit, okay. So I can do some manual annotations myself. So I can take this circular thing and I can zoom in and I can do this, like this, like this. You can zoom out a bit. You can go to these ones and this one, right? And then maybe I'm curious, or I want to tell my colleague about this thing here. So I can zoom in. It's a bit blurry here, but, and I can do something like this. Oh, joke. Hang on. Let me just do this really quickly. Sorry, I just clicked on that. Where's my? I'll do this again, a bit slower. Yeah. Okay. So I can put some text. So you can put some quick text here if you want to, and say, what is this aggregation? So this is a sort of quick kind of manual annotation of your image that you can then save and you can export. You can export this image as a PNG file, so then it will be exported with all of the annotations. You can also export it as a TIFF image and it will be just a raw image without the annotations. But the raw image will contain some of these properties. So with each image that you take, you have all of the metadata saved. So it tells you the settings of the microscope during the acquisition of the image, the camera settings, the session settings, and so on. So you will have all of this information. It will remain with the image. You can plot this as well. So you can click plot displayed. And you can choose from the drop down menu the thing. So you can say, okay, so I want to plot my diameter versus my area, and then you can sort of look at it here. And when you click on this, so this is a very sort of living graph. So each thing that you click on, you will see here. So it will take you back to your image. And if this is us just looking at one image, but we can just as well look at, 100 images and plot a lot more particles on here. And I will show that to you in a minute. You can also go into your plotted particles here, and you can assign classes to these things. So this one here, I can say this is AAV. This is AAV. This one, this one here is I'm going to assign class. This one is a broken one, and this one was also a broken one. Assign class AAV broken. My lips here, I thought this was maybe an aggregate. This one I'll just do as an AAV as well, right? So now when I go back to my plot, I can assign classes for this and have them here so that you can sort of see them in a different way. This is a scatter plot, so you can do a quick box plot or a bar plot. And you can just export or save this graph and throw it in together with this. So this is a sort of quick kind of take a quick image, do a quick manual annotation. Maybe with AAVs, they're not so fun because they don't have as many sort of structural details and so on. This is, I can show you an example. This is a lentiviral vector image taken on this TEM, and there maybe we could do some more kind of measuring and annotations of spikes and so on, and of different particles, and in the same way also then plot it here and export it. So I will go back into my imaging panel. So that was an image that I took. I can also take it in inside different magnification and different field of view and save that and have this as my sort of quick, using the MiniTEM almost as a light microscope, because I think it kind of potentially can serve two purposes. So you can use it to run big analysis on it, but you can potentially also use the system just to have a quick look and check how your sample is, how it looks, almost like, yeah, when you're looking at your cells and you're splitting them and you're going in and you're checking that they look okay. The idea, I think, is to also be able to do that. And if you want to annotate the image, you can do that. And if you want to draw something to the attention of your colleague by doing this, you can also do it. And of course, all of this data that we have can be exported. Just one last quick thing is you can export this data into Excel and plot it further if you so wish. But taking images one by one might not be what you want, and you might want to get a lot of images and do some statistics on this. So the way that we do that is we go into something called our fourth tab, which is called workflows. So workflows is the place where we run different scripts and different instructions on how to detect our images, but it also is where we control screenings and where we can take multiple images. So I will start taking some images now. So I'm looking here and I'm pretty happy with this area, but you know what, we can also just look around because maybe it's a bit boring just to stay on the same area. So we were on waypoint 1 here that we were happy with, right? But I said we could just as well go to a different waypoint, so, or we can maybe visit this area. So I'm going to click here. This is number 3, and I will say move the stage to the selected waypoint. And it's going to go, now it's right, actually the microphone is right next to the microscope, so you can hear the stage moving. Right now, actually, I was pretty zoomed in when I'm moving across the grid, and that might make it a bit slower. Perhaps I should have just zoomed out and then it could have maybe gone a little bit faster, because now it's actually in high magnification, so that's fine. Right, so this was the wonderful area that I chose, but I can zoom out and see, wait, is it really that bad? Am I on a grid square? Probably am. I'm gonna zoom out even more. I was. And here, we're seeing that actually this area, the staining is maybe not as good as an area where we were. You can see that there is a difference. There is some drying of the grid, and there's also some beam damage. So I would probably say this would not be a great kind of grid to image. Of course, you can still see your particles here, and if you really sort of zoom in, you can probably still take some manual images, but this would not be an area where I would do my screening. So you can see them here. So I can just to save time, I can say, well, look, you know what, I'm going to go back to number 1. I was happy there. And to make the journey a bit faster, I'm going to zoom out. And I will click move stage to the selected waypoint. And now I'm zoomed out, so we can have a look, get a sense of, there's a broken grid. It really isn't the best grid, but it's okay. But I guess it also kind of shows the importance of, you know, you need to kind of check more than one area of the grid. And I know in some instances, there's only been, let's say, you know, 5 or 10% that looked okay, but they contained, and that was not for sort of generating statistics, but that was just sort of for pure imaging, for like writing a paper. And they were able to take a bunch of images just in that specific region. I think when you're working with things also like thin sections, you want to find where your thin section, where it sits on the grid, and you might want to look around a bit. If the person that has prepared it might have placed it sort of on a bit of an angle, it might not be so quick and so easy to find immediately. So doing a quick kind of putting waypoints out and taking even a zoomed out image will help you to assess where you want to be doing your imaging. But so we are happy with this area, and I'm going to go to my workflow and I'm going to tell it to take images in this area automatically. So I will click create a new workflow, and the name of this will be take several images, aka screening, but that's fine. This is what I'm going to call it. And I will click here and I'll go to my modules. This might be a bit hard to see. I'm really sorry that this looks so small, but what we have are different tabs or different modules. So we have an input, output, transformation. These are sort of image analysis tools, segmentation, classification, and measurement. And when you create a workflow, you always have to have an input of something. whether it's an image that you have already acquired or whether you want to do a screening, what we will do now. And then your output will be the place where you want to save it. And that's what I'm going to do. So I'm going to take my screening box and I will pop it here. And look, I can actually zoom in now so you guys can, you can hopefully see this better, right? So this is my screening module. And in my screening module, it asks me a few questions. So it says, do you want to use the current magnification? And I do, that's fine. If I don't, I can just untick it and then I get an option of choosing a magnification that I want to take. Then it asks you, do you want me to do autofocus on this? You can decide if you want to do that or not. Here is some information about how much separation you want between the images. So if you take several images, you can also potentially stitch the images together to get a better overview of that specific area. And then you also have something called the dark image rejection. So you can decide this, but you can reject images that are too dark or if they're close to a grid square. The region width is the one that tells you how many images you want to acquire. So right now I have 5. That means I'm taking 5 by 5 images. So that's going to be 25 images in that waypoint. The current position you can choose is either the position that you're in, or you can also choose your closest waypoint or one of the waypoints that you have put. So I can choose fine, closest waypoint. And then I will put in my output. So my output here will be that I want to save the image. So I'll put this here, and by connecting them together, we have created our first workflow. So here it says, you know, do you want to do a histogram modification? Yes, sure. And where do you want to save your, what layer do you want to save your analysis? I didn't really talk to you guys about layers, but in each image, we can use 5 different layers. So we can run different types of analysis on each of these layers. So you can choose where you want to save this. There is no analysis done right now, but if you would run an analysis, that would actually be slotted right here in the middle between the image coming in and the output. You would potentially want to save that, but that will be clearer when I show you your workflow. So I'm just going to run this, and I will, to save time, I'm going to take, let's see, yeah, no, I might, this, oh, sorry, that. So I'm going to take 4 images just really quickly, and I'm going to press start. Yep. So we should be able to see here the preview of our image, as well as the log here in the bottom. So I'm just going to try to make this a bit bigger. So for all of these modules, you can always go to the help panel, and it will give you a description of the modules and what you want to put in and what you have. So this is an aid so that you can get the information of what you want to set. Ah, I was zoomed out. I was zoomed out. That's okay. I'll let it finish taking these. Okay, so it took the images, but actually that was not the magnification that I wanted. So I will go and actually I will move a bit further to the middle of the grid like this, so I don't end up close to the grid square. And I will go to the field of view that I like, this 1500. Right, okay, so this is actually what I want. So I'm going to go back. And I'm going to say, yes, sorry, please use the current magnification, and let's just do this again, and I will press play, and hopefully I get the images that I want. And I'm looking here at the preview to see my image, and there it's come in. How much more time do I have? 20 minutes, okay.
That's with Q&A.
Yeah. Oh, with Q&A. How much time do I have, Salome, for the, 20 minutes total? Okay. Give me like 8 more minutes of this, 8, 10, and then I'll be done. So I'm taking the images. Are there any more questions? No, okay. And you can see here in the bottom, this is telling us the progress of our workflow. So now the images have been taken, I can go into my analysis, and here they are. These are our images that we took. One sec, Emily, this is looking, I just need to, I'm trying to make sure that you guys can see this is best way possible. So there we go. And these are the images. So we can, so now we have acquired images. They are on layer 1, and we can also add tags to our images, and I'll show you why the tags are valuable. So if you are on an image, you can click this button here, and that's a tag. So you can call this something that you want to refer to. So this one I will call session A. Great names here. There you go. And this one I can also call this one session A. This one I will call B. All right, it doesn't really matter what you want to call it, but what I want to show you is, so now we're actually looking at all of the images, but if you click this funnel here, you can choose what you want to look at. So you're not always looking at everything. So you can choose to look at image tags. So if I just want to look at my session A images, I just click here. You can also look at this based on the waypoints that you have taken or the magnification, so that you're not looking at all the magnifications at once. You can decide what you want to filter. And this filter can also be applied to what you want to analyze in your workflow, which is what I will get to now. So we have now taken some images, and we can take, you know, this was taking 4 images, but we could just as well take 100 images. When we normally do a project, we take 5 different waypoints on a grid, and we take 25 images around each waypoint. So we end up with 125 images that we feel is statistically a good sort of range to be in. Then we get really nice representative data of our grid and of our sample as well. And we generate, normally you can see in one image in this sort of field of view, we generate a couple of hundred of particle detections. So from 125 images, you get thousands of points that you can really look at and try to really dissect your sample more. But I will now go to show you then an image analysis and how you can run an image analysis. So this was my screening, just a quick take and save. But I can show you this as well. So this is a workflow that I have pre-prepared. It's a bit bigger. There are more things happening here. But if you look, again, it's a green box here. So there is an image coming in here, and then we are doing a bunch of image analysis, smoothing out the image, defining the edges, calculating pixel values, setting thresholds, and so on. And ultimately, this kind of funnels down into the classification. So we basically give it instructions on what to look for. Look for something this size, this shape, this is the darkness, and then you come down to, and I want you to classify this as, in this instance, it's aggregate. And then here it says, and I want you to classify this instead. If you have your particles and they're close together, you can define how close a cluster should be. So you can tell it a value that if it's X number of pixels, it is not a cluster. If it's like this, it's a cluster. You can separate if you have a cluster of 2 or a cluster of 3. But this is just a regular sort of clusters. And then we're also looking at intact particles and broken particles. So this is what this entire workflow does, and it saves the analysis here in the bottom. So we're going to run, maybe we can run the images that we have just taken on this and see how it behaves. So I will use, I'm going to zoom this in even more. I hope you guys can see this well now. I'm going to use my image filter, and I will go to my tags, and I will choose, let's say, untick this and use my session A images. That's 2 images. And it will tell me which layer do you want me to run this analysis on. Actually, it doesn't matter because we haven't done any analysis on those images. So we could choose any of the 5 layers. I'm going to choose one. And here in the bottom, I'm going to go down and it's going to save it. The image will be the same image, but it will save it on a different layer, so layer 5. If you want to, you can save it as a brand new sort of image with that, but we can save it just as the analysis. So we're ready to go, and I'm going to press play, and I'm going to make this big here because I want you to see it, because sometimes it goes really fast, and it does. So now it's running through our workflow, and the nice thing is that we can see what it's doing. So this one that's grayed out, that's the one that is running right now, and you can see the progress happening here. Some of these are more sort of labor intensive for the software and can take a little bit longer. Other ones, they just swish by. Yeah, you see these ones, you don't even, so I just kind of leave it. There we go. So that's the first image done, and now it's running the second image. Again, you see this one takes a little bit longer. It's called RDP segmentation. It basically looks for round particles of a certain size. And while it's doing this, you don't have to wait. You can go straight into analysis, and we can look at our session A, and we can look at the layer. And there is our analysis, or there is our image. And we can look at this a bit closer. So what can we see here? We can see that, can you guys see this well? I'm going to maybe zoom in a little bit as well. I'm going to make this like this. So it's detected this aggregate here. It's divided up into two. It's not really captured this one. And I wonder if it's perhaps because we have put a size range of the aggregate can only be of a certain size. So one of the things that I have a lot of discussions with our collaborators and clients are, you know, what do you define as debris? What do you define as an aggregate? How close do you want a cluster to be? And do you want information on if your aggregate is made up of your particles that are just kind of stuck together, or and sort of slightly broken, or if it's made of, let's say, host cell proteins or some other kind of junk. And this is something that we can work with you guys and look at this together and try to kind of see how best do we classify your data. But we're seeing that it's actually picked up most of the particles. It ignores these ones here if it's not that it sees the full one. The ones here in the edges are actually removed. You can have a look at what it has classified as internally stained, and that is also something that we can adjust. So here it felt like this was internally stained. Maybe that's slightly on the pessimistic side, and we can change this threshold of this in the pixels. So here, for example, we have a cluster of 4 intact particles and 1 broken particle. We can plot this data again. So this is what we get for this image. As I said, again, we can do a quick scatter plot. We can click here and say, well, what is this one? Is this correct? Is this my biggest aggregate? And we can play around quite a lot. There are a lot of functions and things that we can use to measure and to separate our particles. Again, if you click on this particle and you want to know where it's come from, you can go to your quiet areas, and if you've taken, this is from one waypoint, but actually if you zoom in, you will see right now we have 2 images, and it will give you a sort of a thumbnail of the image, and it will tell you where this particular particle, or where that value, where on the grid that is. So you can really trace back exactly where you were. And for the statistics, you can run a statistics on this, so you can analyze this, and you can get, this was just an example, we can look at the mean, the standard deviation, the different percentiles, and that is also something that is baked in here. And as I explained previously, you can always export the data if you feel like you're more used to using Excel to begin with. That's fine, and you can do that, and you can use the plotting features over there. And again, all of the information is here, and you can look at everything that you click at will be in this image. So this was the circle, and this is this information. To sum up, because we're almost out of time, I just want to run a CNN for you. So this is a workflow. As you saw, it was quite big. It didn't take very long to run the analysis, but we can do this actually potentially even faster. So this is my CNN module. As you see, it's a lot smaller, not just because I'm zooming it out, but it's actually it's a lot more of a simple process. You are putting in your image here. You are looking at you put in this CNN detection here, and you have your different classification. I've just added a little refined particle thing here underneath. It's just that it makes the shapes nice and round, just for sort of visually, it's just easier to look at. And then I'm going to save it, and we can run. Let's see, we can run this one just to show you sort of how quick it is. So I'm just going to run it on one image now, and I'm going to press play. And you can see here it's doing the CNN detection. That's here. It's doing the refined particles so that it makes these now round. And that's it. And we can look at this here again, B. And we go to the layer where we had saved this. I think it was 2. And we can look at our detection here as well. And I found this one. I think I don't have clusters in this one, so it detected this one. I think maybe these were more elongated, but you can definitely play around with this, and you can add more features to it as you're moving along. But looking at this, I think CNN doesn't feel as valuable as it could be, but it really is for us, because when we look at pleomorphic particles, giving it clear instructions on the size and the description of the particles that we look at is a lot more difficult. And that's why I wanted to show you, to kind of finish up with 2 things. So I want to show you this one. So I showed you this before, but these are lentiviral vectors, and lentiviral vectors are pleomorphic, so they will change their shape, and their size can vary. And it makes it hard for us to do the classification and the detection on them. So I will just run our lentiviral vector CNN on this. So I will use my image tag, and I will run my lentiviral vector, and I'm going to press play. And if you look here, that's it. So that's the sort of segmentation that is done. And you can see here that it would have been very difficult for us to do this manually, to tell it this information. And as well, as I was looking at this grid that I was going to show you guys, I also realized that it's a pretty uniform and quite boring grid, and maybe it's not really showing how valuable it can be. So this is actually another AAV image from another grid that I have, and if you can see, there are a lot more broken particles here. We're seeing a lot of variation in our sample, and we can, as I said, we can plot this, and I think the data here is probably more interesting to look at, and then you can say, okay, which is my biggest cluster? Is this correct? Okay, you know, what's my which of my particles has got the highest interior variance, and is this something that I should think about or be concerned about? So hopefully I have conveyed to you the use of this, showing you just a few different examples of what we can use for imaging and the kind of analysis that we can do, and how we can export this data, how we can take images, although these are maybe, as I said, not maybe the most exciting ones to look at. I think looking at your viral vectors, which ones are broken, which ones are not, what's the ratio of this, and looking at what is your debris, what is your background, how does that vary in different purification steps, or if you're freeze thawing your lentiviral vectors, what happens to the spikes, what happens to the spike shedding there. You can do quite a lot of analysis on this, and I think in VaxHub and all the affiliates of VaxHub are doing some really exciting and great projects, working on fantastic and really fun particles. As a virologist, I love to look at and measure, and you know, I hope that I've been able to convey that this can be used for other applications, not just AAV. And as Salome mentioned, you know, we could possibly do a follow-up webinar where we can look at more specific cases. I see there is a question.
Yes, there is a question from
Yeah.
Ellie.
I can stop now.
Okay. So before we start the questions, thank you, Saba, for condensing in such a good way all this information. And thank you, the invitees, for raising the hand or typing the question just to keep it organized. Please, Ellie, go ahead with your question.
First of all, thank you very much, Saba, for what you've done and your colleagues. Very interesting. It's not always the easiest thing to go through all those layers and make them clear. So I actually understood, and that's a good sign.
Yay!
If I can understand it, I think a lot of people can too. Now, the question I had was a small question. You showed a particle size distribution, and it had a sort of a bump. Effectively, I was wondering whether how easy it is to deconvolute this into different peaks. I don't know whether you know which one I'm talking about. The one which is the particle size distribution, where it shows that it looks pretty straightforward, but if you look at it closely, at least on one side, which is on my right, on the right-hand side, it has a potential second hidden peak. Not this one, you know, the one which you showed much earlier on. The AAV.
This one?
No, go back to the very original, the AAV one, not lentiviral, the AAV.
This is AAV, but I can show you. Yeah, let me go to that session A one, maybe. The ones that I had run the analysis on. Let's see.
Yeah. I don't know whether you saved it or not.
Oh, I did. Hang on, it's here. There we go. What displayed? It saves it. It all stays in the project. So when you close the project, it won't let you close it without saving it. You mean like this one?
Well, well, I can't see the detail, but you didn't show this one. The one which actually was a genuine. I think it was the raw data. Well, what I consider as raw data rather than a distribution. The raw distribution of the particle. It has a peak and then it has a shoulder.
Ellie, was that the original comparing it to the DLS?
The very original one, the very first one you showed.
Right, in the presentation.
Yeah, right at the bit. But I mean, you had done the actual measurements, but it was not actually the distribution wasn't like this. It was just a genuine graph.
Yeah, yeah. The one with the DLS overlay. Yes.
Yeah.
Yes. I mean, this is the thing, right? So when you have, so that one came from a comparison that we did with the DLS with the nanosite one. So that information was done by some colleagues and it's actually on our website. But I think it's quite a fun thing, because sometimes what we can see is that we get we can see a slight variation in the size distribution of some of these particles that we often perceive to be to have a very uniform size, and it's something that is maybe quite difficult to pick up when you're looking at 1 or 2 particles, but when you're looking at 40,000 or 50,000 particles, you could potentially pull that apart and look at it further. And I would maybe say that you would want to do it in VIA so that you can click on those specific ones in the graph and really look at, wait, what is happening? Is there something that is it something that is an artifact, or is it actually, you know, a subpopulation of something? Is it that you're seeing something where you've got different serotypes mixed together, or different clades and they are doing different things? Or is it that actually some of these are coming together, or they're stuck together and they're behaving, but this kind of sticking together of coming together as duplets or triplets is significant enough that it will provide you with a second bump or something.
Yeah, exactly, because I find that data with the shoulder obviously becomes important. That means that you have to deconvolute it further to get more information. And I just wanted to make sure that with your system, that is something that is doable, that we can then focus, so that once I actually, sorry, if something comes up all the time on my screen, I apologize. So that if we get convolute this and get to the shoulder, we can relate that shoulder back to the image and say, this is what was on the image, which was then translated to a shoulder.
Yeah.
Okay.
Yeah,
if
absolutely.
can do that, that would be very useful.
Yeah, exactly. And this is, for me, this is what I kind of, you know, one of my real favorite things is you can take something here that is an outlier or a subpopulation like this, and you can actually really just go into it further and further and actually find out here and here, what, you know, what was this? Is this real? Is this something that is of value? So yeah, I would say, and if we have some samples like this, let us run it here and let us really dissect it effectively.
Okay. Thank you very much.
No problem. Sorry that I didn't know which one you meant. Thank you for helping out.
Steffi, you can ask your question, please.
Yes, thank you so much, Sarah. I really enjoyed that presentation. So I work at UCL. In my lab, we do quite a lot of EM. We're looking at different types of protein nanoparticles, tubular structures, and microcompartments. And what we struggle with a lot, well, not time-wise, is the data analysis. So we also do a lot of sectioning through bacterial cells. And I was wondering if your analysis, your analytical platform, can also deal with sections through cells. I know you showed some sections, but in terms of the analysis, I wonder, because it's a little bit, I guess, it's more crowded. There's lots of things going on inside the cell. So this takes a long time for us to do. And I mean, this looks like if that would work on sections, that'd be fantastic. I'd be really interesting to talk to you more about that.
Yeah, we like sections. They are a little bit more challenging, but it's actually something that we do quite regularly. The actual, you know, the process to actually generate the grid or, you know, to generate the sample is actually the one that is the most time consuming. We have 3 or 4 different applications that we can specifically use for this. So do you remember when I told you that you can sort of, we can stitch together images to give you a better overview. So if we just want to talk about looking at these thin sections, the first thing you want is to get, you know, not a choppy image, but you want to have something that you can really zoom out on and look at. And we can stitch the images together. And we also use different things like template matching and so on to analyze these images in the thin sections. And by reducing the spread of our electron beam, if you have it on not a single slot grid support, it works pretty nicely. Of course, the only thing is then that you do have some grid squares coming in, but sometimes some of these hexagonal grids can help you to reduce the interference of the grid support. But we have clients as well that have got the MiniTEM system specifically for thin section analysis. So something that we are quite comfortable with. So yeah, I'll be happy to have a conversation with you and look at this further with more specific examples.
Fantastic. I can send you an email. I have one more little question, and that is, I notice, does your sample holder hold only one grid? Because this is another time-consuming bit where you wait for the vacuum to come, you know, to close and so on.
It does. It does. The plan is that it will have a 3-grid holder, but that is the plan. We understand that it is time-consuming, and I think that is probably one of the things that will be taking kind of the most time is going through this the sample exchange. But yes, unfortunately, at the moment we only have one.
Okay. Thank you.
No problem. Thank you.
Thank you, Steffi. Any more questions? Adil, Steve?
Hi, thanks a lot, Saba. That was really, really great. What I really enjoyed was actually to see the how you chose not your best grid. to show how really actually it's the sample preparation, the grid preparation. Once you optimize that, then actually you know optimize the sort of the amount of sample you're putting on and the conditions for staining. So it's good to know that that's still always the case. One of the things I'm interested in is obviously we're making various constructs, different particles, virus-like particles, which may have quite pleomorphic representations. And I think ultimately what we would like to know is whether those things are functional. So we would do morphology, but then linking that to some type of functional output, so immunological or antigenic. So the question I had was really based on the viral vectored stuff you've done and showing the difference in AAVs. How do you link that data and the analysis to the function of those viral vectors?
So we always like to get as much information from the user as possible to look at these kind of links. It's more that they come with us with other techniques that they have used to say, okay, you know what, here is a purification that I'm doing, but I'm getting 2 peaks, but these 2 peaks behave exactly the same, but they come out at slightly different size. And I want to know, I've tried different things, but I want to know, is there something here that is different? Is the size distribution of these 2 peaks different? Whether we can then correlate it back to a function or not, that is sort of up for discussion. But one of the things that we think is fun and sort of valuable is to get enough data. You know, when we talk about 100,000, 20,000, tens of thousands, and getting really heavy statistics to start seeing any potential subtle variations, but variations that are coming up frequently that can be attributed to whether it is that the samples are slightly more elongated, have got something different in terms of their spike coverage and so on. So it's VLPs and pleomorphic particles and the analysis that we can do and how we can chop it up. I hope with discussion with you guys can help us to kind of answer the questions. But I guess you guys would have the questions and we would just give you the statistics and the data and see that this is what we are seeing. And one of the things that is also important to try if you're working with new particles and so on is to try different stains, try different things to make sure that we are not getting any artifacts that are not attributed to your sample in particular. So sometimes some of these VLPs or lentiviral vectors, they don't behave the same way in lower pH because you use that like endocytosis, so they would potentially lose their spikes. So we want to make sure that also the data that you are getting is as accurate as possible. I don't know if that really answered your question, but I hope it sort of did. But if not, I'm happy to have a conversation with you and show you how we have worked with VLPs.
Thank you very much. No, I think it's a really great, to make electron microscopy more accessible. So I'm hoping, yeah, with VaxHub we get opportunity to put some exciting examples through this system.
Yeah, me too, absolutely.
Thank you so much, Adil. Is there any other question? No?
Can I just say one last thing? I actually forgot, is that if you connect this up, you can run this remotely. It's kind of one little thing. So I do a lot of the image acquisition from home, just so you know. So if there are I'm just thinking in terms of lockdowns and so on, this microscope can be run remotely with the image analysis as well. So we can find different ways of you to still interact very much with us, whether you want to just take the images on, talk to Salome, take the images and then we do the image analysis together, or if we want to do the imaging remotely together, that is also possible.
Steve?
Yeah, I was just going to sort of make sure that everyone sort of got the message on that. Saba's point there is that they can also work with images that any TIFF image file, isn't it? That's correct, Saba?
Yeah, so we like to have OME TIFF files, which is sort of open microscopic environment TIFF files. We've imported. We have done this previously, so we can work with most TIFF files. Yeah.
Yeah, so I mean, if people have got images already, then they can just send them over. Yeah.
Exactly, and that would be really, really quick. We have got a quick way of also receiving the images, and we can convert them quickly, and we can look at them immediately.
Any more questions? Okay, so if there are no more questions, I will finish the session now. Thank you all for attending, and thank you, Saba, for all the information that you gave us, and I hope that we will receive good feedback and new opportunities to collaborate.
Absolutely, me too. It's been a real pleasure hearing myself talk for an hour and a half. No, but thank you so much for your questions, and that was really good. And I really hope I haven't, like, you know, I hope it's not been too much. And I'm really sorry if the text came out a bit small. And I look forward to having a follow-up session if possible. Then I'll make sure that we can look at some of these images even bigger. So yes, thank you very much. I'm looking forward to all the collaborations and contacts. Thank you, and thank you, VaxHub and Salome and all of you guys for giving us the opportunity to present. Thank you.
Welcome. Okay, so bye-bye, everyone. See you all soon. Keep safe.
Absolutely.
Thanks,
Bye-bye, everyone. Bye.
Thank you, everyone.